University of California Cooperative Extension and University of California Davis Veterinary Medicine is bringing to you a recording from their March 2022 webinar series. This series was co-hosted by University of California Cooperative Extension Advisors Tracy Schohr, Grace Woodmansee, Rebecca Ozeran, and Specialist Dr. Gaby Maier. Additional resources from this webinar and other CattleHealth videos can be found at ucanr.edu/sites/rangelands/CattleHealth. This session is focused on why did it die and submitting samples to the California Animal Health and Food Safety Lab with featured speakers Dr. Gaby Maier, Cooperative Extension Specialist in Veterinary Medicine at UC Davis, and Dr. Katherine Watson, Pathologist at UC Davis.
We are talking today about submissions to the CAHFS lab and some of the commonly seen diseases that are diagnosed there and how to take advantage of the diagnostic lab the best. So the subtitle, Why Did It Die, is a little bit tongue-in-cheek, but that's kind of the goal for tonight. So again, I'm Gaby Maier. I'm the extension veterinarian for beef cattle. And I certainly make a lot of use of the diagnostic lab, especially for the research trials that I have. But there is the diagnostic lab is just a great and wonderful resource. And so hopefully we can convey that to you tonight. So as an animal owner, you typically work with your veterinarian when there is an animal health problem. And so your veterinarian may then, if an animal dies, may come and do a necropsy in the field and then submit samples to the lab or submit the animal for you. But you can also, as an animal owner, submit to the CAHFS lab yourself. And a lot of animal owners do that. So this may be because your veterinarian is not available at that time, or you just don't have the time to wait. You want to get an answer right away. So at the CAHFS lab, a necropsy is then performed and they generate reports. And there is actually multiple reports typically on a case. So when they first open up the carcass and look at the organs and everything, they may have a preliminary idea of what could be the case, and they will give you a report of that first step. But then there are follow-up tests. So there may be bacteriology or virology or whatever test they think is necessary will be done. And they can take some time. And so you will get follow-up reports until everything is concluded, and then hopefully a diagnosis is made. And then based on that diagnosis, you can use that information to then instigate maybe treatment for animals that are alive and that are sick, or you can change your management in order for this disease not to occur in the future. So we're talking now first about the mechanics of a submission to the lab, because it's really important that you as the submitter do your part in making this a success. So this is the web page of the CAHFS lab, and when you go there, you can download the submission form, and you should do that in any case when you submit something there. So you find under Submit Specimens, you go to Submission Forms, and that will download the correct form. And this is what it looks like. And let me just activate my pointer, my laser pointer. So, as I said, it's always best to work with your veterinarian anytime something goes wrong with your herd. But if you submit yourself, you can keep your veterinarian in the loop by just adding his contact or her contact information in this field. And so then they will get the results as well. So they're already in the loop. So this is the business information, so to speak. And then we have case information where you fill in, what type of animal it is, you fill in some history, and we'll talk about that in more detail. You should, if possible, fill in this information as well, where the animal is located, what production class it is, if it's beef or dairy. And then also the animal and test information. So if it is a dead animal that you want to submit for necropsy, you would put in the animal ID, what breed it is, male or female, the age, and it has to come with, don't just write 2, is it 2 months, is it 2 years? So what is it? And then it would be 1 carcass for necropsy, is what you would fill in, and they do request that you sign this because this is basically the way you order these tests. So where is the CAHFS lab located? So there's 4 locations. There's one here in Davis, where we are, where I am also. There's one in Turlock, Tulare, and San Bernardino. They all take beef submissions or cattle submissions, except for the one in Turlock. The one in Turlock at the moment only does avian necropsies. That's because the poultry industry is in that area, so they have specialized on that. But they are in the process of building a new facility there, and then once that is finished, they will accept all species. You can submit, though, other samples. So if you want to submit blood samples for, let's say, for trace mineral testing or something else from cattle, you can submit that in person to the Turlock lab, and they will send it to the corresponding lab that does that performs that test. And the same is true for the other lab. So if a particular lab doesn't perform a particular test, they will send it on to another lab that does. And so this is just a slide that shows the versatility and the overall mission of the CAHFS lab. So they provide many different services. So there is some animal disease surveillance. And so that is actually one of the big purposes why we have the CAHFS lab, and why the necropsies that are done there are at a really good price. So we'll go over the price structure in a little bit, but I can guarantee you it is a really good deal for what you get, the expertise and all the testing that you get. It is very, very good, a very good price. So one of the reasons why it is subsidized by the state is because they want submissions so that we know what is going on, so that we don't miss a lot of diseases that we may not be aware of. So it is part surveillance, but there's also active surveillance. So for example, when I worked for the California Department of Food and Agriculture a few years ago, and I would go to live bird markets and swab poultry, and that would be tested for avian influenza. So there is some active surveillance for foreign animal diseases. They provide services for health management. So for example, some of the labs have a milk quality lab that cultures milk for mastitis pathogens, food safety, so testing for things like salmonella, new knowledge. So there is research that is always going on with the scientists at the CAHFS lab, and in conjunction with other scientists at UC Davis. Equine health, so they have a laboratory that tests equine athletes for performance-enhancing drugs, and of course also public health service by diagnosing diseases that could also be zoonotic. So there is a wide variety of services that we get from this lab system. So when you submit something for necropsy, what's preferred is the whole animal, the whole carcass, because that will give them the best option to open it up and recognize things that are abnormal, and then take the best sample to do further testing. But sometimes that's not possible because you live far away. You just can't, you can't do it. You don't have the time, or for whatever reason, you cannot bring the whole carcass there. So if that's the case, then you can still submit samples, and we will show you how to do that, or we will go into more detail of how to go about that. But it's recommended that you first get in touch with one of the pathologists. So give them a call and talk the case through with them so that they can give you advice on what kinds of samples are best to submit for your particular case. There is here on the right is the CAHFS contact information, and you can look that up online. The normal business hours are from 8 to 5, so it's best to call during those times. If you have an urgent case that occurs after hours, there is a possibility to call and then leave a message for the pathologist on call. So typically until 9 p.m. they can be reached. And they can take up to 2 hours. So you can leave a message, but within 2 hours, you should expect a call back. So when you submit, a good history is really important. So, when you submit a carcass and the entire history was sick for 2 days and then died, that's not too helpful. So you really want to include as much as possible, as much detail as possible, because every little thing can be a clue and can lead to a diagnosis, can be important in coming to a diagnosis. So was it a sudden death, or was the animal sick for a while? Was it coughing? Was it not eating? Was it lying down during that time? Was there any discharge from the nose, from the vulva? Did it have diarrhea? All those things can be important. Also, were any treatments given to that animal? So in particular, antibiotics, because if antibiotics were given, and then during necropsy, tissues are being cultured for bacteria, maybe nothing grows because the antibiotics did kill those bacteria. The animal may have still died because the disease process has gone on for too long. But that could be an explanation for why certain bacteria couldn't be grown from the tissues. Also, the vaccination history can be important, whether the animal was recently dewormed or not. And then also if any other similar cases have occurred in the herd before. And then since we all have cell phones with cameras, it's so simple to take pictures. So it's also recommended to take a picture of the carcass, how you found it in the field. Maybe there are some clues, maybe the animal was thrashing or not. So that could be a piece of information that's important. If you see any plants that you think could be toxic and that the animal may have eaten, take a close-up picture. So, you don't want to take a picture of the whole meadow. You want to get really close and make sure it's in focus so that the toxicologist may be able to recognize a toxic plant. You could also take a handful of those plants and put it in a plastic bag and submit it together with the carcass. So this is again the submission form, and this is the second page. And I just wanted to show you this to give you an idea how much room you have to write. So this is all for history. So there is really no excuse in not filling it in. This form can be filled in on the computer. So when you download it, all those bluish areas, you can type in those. So, it makes it easier. I mean, some people prefer that. Some people may want to print it out and fill it out by hand. So when it comes to toxins, one of the things that you need to realize is that unfortunately there is no toxin panel. So we have other panels, like we have abortion panels where we can test for a variety of different pathogens that cause abortions, or diarrhea panels. But when it comes to toxins, they all have to be tested for individually. So you can't just submit some feed and say, oh, just run a toxin panel. I think maybe it was a toxicity. You have to really know what you want to test for. And so, for example, this picture, shows some, nasty looking algae. And so if you find animals that are dead next to a pond that looks like this, that may give you a clue that maybe this is the cause. And so a sample of this water could be submitted and that could be tested for blue-green algae toxins. But that is one specific test. Another example could be if you have a new bag of feed that contains monensin, and the animal shows signs of monensin toxicity or lesions that are that could be a monensin toxicity. That in that case, you may want to test for monensin. But all these tests, they're unfortunately many of them are not included with the regular submission fee. And so if you just nilly-willy test for anything, then it could add up a lot. And then in the end, maybe you end up with no result and you've spent a lot of money for nothing. So you really want to have a specific idea for what you want to test for. And of course, you get to that point together with your veterinarian and the pathologist. And so when it comes to cost, the cost of the regular necropsy with most of the follow-up testing would be $140 for an animal over 3 months of age. And there's a $50 carcass disposal fee. And then for 3 small calves, so up to 3 months of age, it's $135. So, but how do you collect those samples if you cannot submit the entire carcass? So we actually made a video on how to do this, and I will show you a little sneak preview. It's not quite finished yet, but it will be available on YouTube. And so you get a little sneak preview tonight. In any case, so this is some tools that you may want to have handy. So there are garden shears, and you need those to cut the ribs. A sharp knife that's dedicated for that purpose, so that does not go back into the kitchen. And then just some regular scissors, some tweezers, and a couple syringes, needles, and red top tube to collect blood or urine would be good. Also, what you need is some Ziploc bags and some other packing material that I will show you in a minute. But before we get there, I will show you some of the video that we made. And I'm just going to flip through it a little bit. So this is Dr. Kate Watson, and we'll show you again. We're showing you here the tools. And so just as a warning, we are cutting open an animal. So if that's something you don't want to see, then you may want to go and get a drink or something for the next 5 minutes. But if you are not squeamish, then we can show you how that's done. So we'll show, and this, of course, it's narrated, so it has sound. We're showing you how to examine the calf externally, what to look for in the eyes, etc., in the mouth, the tongue, in between the feet, the umbilicus, etc. So all the external parts. And then we'll show you, or Dr. Watson shows, how to open the carcass in the field, how to take a blood sample, so with reflecting both limbs, the skin is then being reflected. The abdomen is opened. The diaphragm is punctured. And now we're opening the rib cage. And this is where you're going to need those garden shears. Here they come. And then we have all the organs labeled so that when you open it, you see what is where. And this would be a good time to take another picture for, that you can then send to the pathologist. And again, we have everything labeled. Show you how to remove what's called the plaque. So that's everything from the tongue down to the respiratory tract and the heart. And I'm just going to skip through this. Shows how to take samples and how to open up the kidneys, how to open up the intestinal tract. We're even taking off the head in case you want to have to submit the head. We're opening up joints. And then this last part shows how to wrap samples from individual organs. So for example, you can use a rectal sleeve and make little compartments and then put a knot in between and then add the next piece. So as I said, this will become available in the next few weeks. Again, this is just a list of all the things that can be done at the CAHFS lab. So necropsy is when the whole carcass is being examined, and then that has to follow up with histopathology. So every organ is then examined under the microscope, and IHC is immunohistochemistry. So that's when those tissues are stained to look for specific pathogens. Bacteriology is looking for bacteria. Toxicology, obviously, for toxins. Serology is doing tests on blood samples to see if the animal has been exposed as an antibody response to pathogens. Parasitology is looking for parasites. Virology for viruses, and molecular biology are tests such as PCR tests that also look for particular pathogens. So again, what you want to submit is kind of a fist-sized piece of every organ. And so this would be an example of a good submission. So everything is nice and clean. It looks fresh. It has the right size. So you don't want to submit, tiny little pieces that may not be good enough to run multiple tests. So sometimes these tissues are used for multiple tests, and that's why it's good to submit a good chunk. It's also when you do this, when you open up the carcass and see something that looks abnormal, where you sample is important. So this blue square here shows the area between the lung. So this is a lung. The lung that looks normal. So this upper part is kind of what a normal lung would look like. And this lower part that looks all reddish, that is abnormal. So that is consolidated lung. That's a pneumonia. And the junction between the normal and the abnormal is where you want to grab your sample that has the best chance of containing the pathogen. But if you grab a sample of just the part that looks really nasty, so to speak, that could be where the pathogens are already dead because they have been festering for a while. And the tissue is so destroyed that it's not really that useful anymore. So when you ship, when you send these samples in, the faster the possible, the fresher the better. So you want to ship it on ice, not frozen, because freezing can destroy the tissue. So if you freeze something and then it thaws, it can destroy some of the tissue structure. So it could be difficult to look at them under the microscope. And you want to add a generous amount of these gel ice packs. So not just one, you really want to pack in as much as possible to make sure everything stays nice and cold. So double bagging in Ziploc bags so that you don't contaminate things with each other. The submission form, also put that in a Ziploc bag so it doesn't get stained with things that could leak from the other bags. And then you can call the lab and ask for a FedEx account number. That way you get a discounted overnight delivery. So they will give you an account number that you can fill into your FedEx form. You will still have to pay yourself for the shipment, but it is discounted, and it is the preferred way to send in samples. And if you have any questions about any of this, again, call them ahead of time, make sure that what you're doing is how they want it. So all of this information is actually summarized in a handout that we made just for this purpose. So it was actually Grace, Dr. Watson, myself, and another pathologist from CAHFS lab. We put something together that kind of summarizes all this information of how to sample in the field and then send it into the lab. And this can be found here, and I'm sure this can be also made available. There are other possibilities of how you can submit this. So obviously bringing the animal yourself, or you can call an animal hauler that will deliver to the lab, or you can use any of these carriers. So Golden State Carrier, FedEx, a UPS, or a USPS. But when you ship it, make sure that it gets there overnight. And then we already talked about this account number. So this is what those ice packs look like. And best is to use a styrofoam shipper so that things get there cool. Saturday delivery is not recommended. So if you send something on a Friday, if you want to send something on a Friday, make sure that, again, that you call ahead of time to make sure that somebody is there to take that. Yeah, so it will be received on a case-by-case basis.
Yeah, so there's, hey, Gabby, there's always somebody there at the Davis CAHS laboratory from 8 to 12 on Saturday. Like the shipping companies like FedEx or Golden State, especially with kind of decreases in the number of people we have working for those companies, they don't always have somebody to deliver them, or they don't deliver them on time, like the shipments are delayed. And so that's where people get into trouble, because then they're not refrigerated for enough time, because then they sit all weekend in the storage facility that's really hot, which is why we don't recommend a Saturday shipment, because it's just less reliable. There is somebody at the CAHFS laboratory there. It's just, it's really hard for the shippers to actually deliver it on time.
Yeah. So you don't want to risk, having those samples be spoiled.
And just as a tip to build on that, like, so if you've ordered vaccines, something to think about when they come in those styrofoam shipping containers. So keep 1 or 2 of them in the back room, so then you wouldn't have to buy that later, and you would already have the supplies to ship them to the CAHFS lab.
Exactly. Good point. All right. So here are some other ideas for how you can make use of the CAHFS lab. So trace mineral testing is something that is often done for herd health. So copper and selenium deficiencies are very common in California. Liver biopsies are the gold standard to test for trace minerals, in the trace minerals, but often it's blood samples that are used. So if you do use blood samples, you need a purple top tube for selenium, and blue top tubes are recommended for trace minerals, because the stoppers in those blue top tubes don't contain zinc. The other ones, the red top tubes, they do, and so the zinc could be inaccurate. And another idea is to test calves for IgG levels. This has to be done within the first week of life. And this could be the case, or that you may want to think about this if you have a lot of calf foot diseases, such as diarrhea or respiratory disease, where you're just struggling with it, and you've kind of tried everything, but you don't really know what else it could be. So failure of passive transfer is something that we typically think about in dairy calves. However, it has been shown that beef calves actually also can often have failure of passive transfer, so where the calf just hasn't ingested enough colostrum. So you could test some of your calves, and again, it has to be within the first week of life, so after 24 hours, but within 7 days of age, and see whether that is actually the case or not, and then make adjustments accordingly. And finally, another way you could take advantage of the lab is by testing of herd additions for some of the chronic diseases that we see. So for example, Johne's disease, bovine leukosis virus, BVD, persistently infected animals or anaplasma. And these are things where you definitely want to talk with your veterinarian about what the risk is in your herd versus the benefits of testing. Because obviously, if you run a lot of these tests, it adds up. Especially if you test everybody for Johne's disease, that can be very expensive, and the test is not always very sensitive in younger animals. So it may not be the best in your situation. So you definitely want to talk about it with your veterinarian if this is something that might be beneficial for your herd. But in any case, if you add animals to your herd, you may want to think about testing them for BVD-PI, persistently infected animals, because that is something that you absolutely don't want to introduce into your herd. And with anaplasma, it's more where a situation where if you are in an endemic area, so if you are in an area that has this disease, has anaplasma, and it's a tick-borne disease, so it's the ticks that spread it. It's usually in the foothills where this occurs, not so much in the valley floor. And if you purchase animals that have not been exposed to this disease and they are then moved to an area where the disease occurs, then it can end up with a disaster where a lot of animals get sick or have abortions or die. And so obviously you want to avoid a situation like that.
Hi, everybody. I am Dr. Kate Watson, and I am a pathologist at the California Animal Health and Food Safety Laboratory. I know somebody asked what CAHFS stands for, and I know that it's easier to say CAHFS, but when we think about it as an entity, it is the California Animal Health and Food Safety Laboratory. It is on the UC Davis campus, but it is funded by the California Department of Food and Agriculture. So it's a slightly separate entity than the UC Davis campus itself. So I'm just going to touch really quick on this one point, that a good quality sample is the best in getting a good quality diagnosis. So we do sometimes get things into the laboratory where there's been significant predation. You can see in this animal, there's a lot of autolysis. It's probably been sitting out in the sun for a long time. In addition to that, there has been predation. So a significant amount of this horse has been preyed upon by scavengers. And that includes most of the internal organs, right? So the chest cavity and the abdominal cavity have collapsed because there's no more internal organs. And so sometimes we get lucky and we're able to give you a diagnosis on these samples. And if it's the only thing you have, we are happy to accept it. But sometimes you also may want to pick and choose, especially if you're having a lot of loss on your in your herd. You may want to try to pick the best samples to send to us so that we have the best chance of getting you a good diagnosis. All right, so on the agenda of diseases that we're going to talk about today, these are some of the things that you guys sent in. I want to thank you for sending in some of the things that you wanted to hear about, because that makes it easier for us in targeting what your interests are. There were a lot of great suggestions out there, and I apologize ahead of time that we weren't able to capture all of those suggestions in the time that we had. But certainly we hope to do another seminar series where maybe we can touch on some of those ideas in the future. So today we're going to talk about bovine respiratory disease. There were a lot of people who wrote in about wanting to hear about that, as well as blackleg, Histophilus somni, or some of you may know that as Haemophilus somnus. So that was the previous name for it. And for some reason, bacteriologists really like to change the name of things to keep us on our toes, and it makes it very confusing, both in the literature, but also as a veterinarian on making sure that we're most up to date. And then we're also going to talk a little bit about bovine viral diarrhea. And lastly, we'll touch a little bit on abortion, but focus in on foothill abortion, which is what people were interested in hearing about. All right, so we're going to start off today with some bovine respiratory disease. But before I go into the actual disease process, we're going to launch another poll for you. I think one of the most difficult things to do when you're doing a field necropsy, which some of you might do if you're kind of more in rural areas and can't get a whole carcass to the CAHFS laboratory, is determining what's a lesion or what's really concerning, or what may be an incidental finding. So in looking at this picture to the right, you can see that there are all of these black spots that are scattered throughout the pink tissue. So if you had to guess, is this concerning or is this incidental, like not important, what would your guess be? So you guys are focusing in on those black areas, and in some animals, this could be concerning. So if you have a gray horse, for example, you might be worried about melanoma, right? So if you don't know, gray horses are really prone to melanoma. They may start kind of in the tail region, and then they can spread to other parts of the body. But this is actually a normal finding in the lungs of multiple species that have black faces. So for instance, you can find it in Holstein cattle. You can also find it in black-faced sheep. And so there's a number of different animals that have that black face that can have melanotic spots or black spots from melanoma, or from, sorry, not melanoma, but from melanocytes within the lung, and it's totally normal and incidental. But when in doubt, sample it, right? Because if it's enough to make you concerned, it's better to sample it and send it in, and then we can tell you whether or not it is it's something that might have caused death, or if it's something that's incidental. And this is where that cell phone picture can be really helpful too, because distribution, whether it's just on the surface of the lung or whether it's deeper down, can be helpful for the pathologist as they're trying to interpret the lesions or what they're seeing when they only get a small part of the animal. So I appreciate everybody who put their answer into the chats. All right, so let's talk a little bit about factors. So don't worry, I'll get to the gruesome pictures. You don't have to be too concerned about that. But first, I'm going to give you a little bit of background on factors that contribute to BRD and why you may be seeing bovine respiratory disease in your herd. So bovine respiratory disease is a leading cause of morbidity and mortality and economic loss in cattle. It is one of the largest health challenges facing the modern day beef cattle industry. Sick beef cattle also grow slower. They develop less efficient feed conversion ratios, and they tend to need additional feed time to reach similar carcass quality of clinically healthy calves. The wide use of vaccines to prevent and treat bovine respiratory disease is a common approach worldwide. And I think Dr. Maier has already talked a little bit previously about vaccination protocols. However, what she may have mentioned in that vaccine series is that we're not actually getting the desired effects of vaccines to protect against bovine respiratory disease, like the level of protection we're not quite achieving yet that we expect. And part of that is because a good herd health program is just as important as vaccination. So vaccination isn't the only part of of managing disease on your farm or in your herd. So cattle that are at risk of bovine respiratory disease, they are being affected by pathogens. So these are some of the triggers, pathogens. Changes in the environment and management, which includes weaning, commingling, so when they're in a new environment, or commingling during transportation, and also dietary changes. This sets them up. So a lot of the pathogens we're going to talk about today, pathogens being both viruses and bacteria, a lot of the bacteria that they have are actually already within their respiratory system. So they're what are called they're like part of their normal bio flora. So when we talk about, probiotics and you're trying to change the flora in your intestines, so we have bacteria that live in our respiratory system. And it's when those bacteria in our immune system are out of sync that we get this disequilibrium, and that results in respiratory disease. All right. So the other thing that factors in are vaccines. So it's really important. I know that, so Gaby mentioned earlier in the talk about how one of the things you can do is send in blood samples of a calf that's less than 1 week old in order to measure immunoglobulins. So those are antibodies that the calf has within the blood system. Those ones that they obtain in the first few days of life come from what's called colostrum. So colostrum is the first milk that the dam produces and that is suckled by the calf. That contains antibodies that help protect that calf until their own immune system can pick up and start actively producing their own antibodies. So you can see in this chart over here, these would be the antibodies that the calf received from the mom. So in the colostrum, that's called passive immunity. Over time, that is going to wane, right? Like the antibodies are going to break down and they're going to be filtered out. So you're going to lose those antibodies over time, but you're going to vaccinate the calf, and then the calf is also going to start making its own antibodies. So this is the calf making its own antibodies to some of the pathogens that may be in the herd. The problem is there is a period of time in which the animal is susceptible to an infection because there's not enough maternal antibodies to protect the calf. And there isn't enough of the animal's own active immunity. So this is called the immunity gap, and there's no protection or not adequate protection. And so they're more susceptible to getting a disease, including bovine respiratory disease. So some people might say, well, we should just vaccinate earlier so that we can narrow that window. And we are trying to optimize those vaccination protocols. But the problem is if you vaccinate too early, those maternal antibodies make the vaccine not very effective. And then if you vaccinate too late, the animals are unprotected for a longer gap period. So that just sets up for why, a lot of the questions that I get when there's a calf that comes into the laboratory that is diagnosed with pneumonia, and then we culture or isolate a virus, they said, well, hey, doc, I've been on a really strict vaccination program. Why is it that I'm affected with this agent or this pathogen, even though I'm doing everything I can to protect that animal? So you may be doing a really great job, but just knowing that there is a period of no protection. All right. So what are you looking for when you're looking for signs of bovine respiratory disease? So this over here on the right-hand side, these are some of the kind of what we call gross pictures of some of the things that you can look for. Here on the left-hand column, these are what should be considered normal. And then these ones are pictures of what is abnormal. This scoring scale that I'm presenting to you the pictures from were actually developed in dairy calves. So it's not validated for beef cattle, but it's a good reference about clinical signs to watch for. So they developed a scoring system to determine if there was a large chance that the animal had bovine respiratory disease. And some of the metrics that they used were whether it had eye discharge, because usually it's you can get upper respiratory signs, and that's from kind of your your sinuses. So like when you get a lot of fluid dripping out of your nose, maybe you have sinusitis, down to your kind of back of your throat and your trachea versus going down into your lungs. You can get nasal discharge. You get ear droop or head tilt. This is because one of the agents that's implicated in bovine respiratory disease is mycoplasma, and it really likes the inner ear, and that throws off your equilibrium. And so then you might tilt your head because you have bacteria and inflammation in that inner ear. So if you've ever had an inner ear infection, you know that that can be really off-putting and can make you feel like you're on that roller coaster because you can't quite balance yourself. So these are all things to look for if you're trying to decide if one of your animals has bovine respiratory disease. So ask yourself, does it have eye discharge, nasal discharge, droopy ears, coughing, difficulty breathing, or a high temperature? All right. All right, so what are some of the causes? And then we'll get to the pictures. All right, so like I said, the pathogens that are implicated in bovine respiratory disease fall into two major categories. You have your viruses, and then we have bacteria. And a lot of times, the reason why we call it bovine respiratory disease complex is because we don't often have one of these agents working alone. Oftentimes, there are multiple agents that are contributing to disease, and that can make it really difficult to tease out which one is the cause because they're oftentimes working together. So some of the viral causes that we think about are bovine herpes virus 1, which is also known as infectious bovine rhinotracheitis virus, or IBR. We also have parainfluenza virus 3, or PI3, bovine respiratory syncytial virus, so BRSV. And the reason why it's called this is because in the lung, it likes to make what are called syncytia. And that is a cell, so it takes the epithelial cells, so all of our mucosal surfaces, like the surface of the trachea, the surfaces of the lung, they're all lined by what we call epithelial cells, and it takes multiple cells and combines them together into one big cell, and that's called a syncytial cell. So that's why this is called bovine respiratory syncytial virus. It does that in the lung. It makes giant cells in the lung. And then we also have bovine viral diarrhea virus, which we'll talk a little bit about later in the talk today. There's a few other viruses that are implicated, but these are kind of the major ones that we think about. Then the bacterial causes, and again, like I said, the bacteriologists, they like to change up the names. So a lot of these will like have it's kind of like the artist formerly known as Prince. Well, we have the bacteria formerly known as these different names. So Mannheimia hemolytica, Pasteurella motacida, Histophilus somni, Mycoplasma bovis, and there are other Mycoplasma species out there, but this is probably our major one. Truparella pyogenes, that one is mostly in calves. We don't really see it in adult cows. And then Biebersenia trihalosii. So this is one that's kind of on the margin. It was actually discovered in Tulare County by Dr. Bieberstein, and it is reported in the literature, but I would say that our major ones are Mannheimia, Pasteurella, Histophilus, and Mycoplasma. All right, so as we think about the diseases that each of these bacteria or viruses cause, they may have a distinct pattern that might give you clues as to the cause of the disease when you're doing a field necropsy, or if you're working with a pathologist, this may be how the pathologist is kind of saying, oh, I think it's this and not this. So first of all, this is the normal lung. You can see how the trachea comes down here, and then it breaks off into primary bronchi, and then these smaller bronchioles, and then it branches again into these smaller airways all the way down into what we call alveoli. And alveoli are like these big sacs, and that's where air exchange takes place. So you can see when we have what we call bronchopneumonia, we have a distribution that tends to be cranial and ventral. And that's because you'll see in a later picture where it's where the CAHFS is opened up. It's at the front of the lung based on how the animal is standing, and the bottom. And that's because bacteria come down the trachea, and bacteria are very large organisms, and they fall out into these first lobes because the bacteria can't make it all the way to these smaller airways in the back. So the bacteria fall out here. So this tends to be more of a bacterial disease. Then you may have a fibrinous bronchopneumonia, or what we call pleuropneumonia. All that means is that on the surface of the lung, you have a substance called fibrin, and fibrin is just an indicator of kind of inflammation and disease. There are some agents that tend to do a pleuritis or a pleuropneumonia more commonly, and that's like Mannheimia. So Mannheimia really likes to do fibrin on the surface of that lung. So it's what we call a really hot pathogen. It does a lot of destruction really fast. That's different from what an interstitial pneumonia is. Sometimes people call it a viral pneumonia. The whole lung is a little bit bigger than normal. And it's because the viruses can make it all the way back into this region that the bacteria couldn't make it into. And then you get a lot of cells in there that make the lung look puffed up, but not like the bronchopneumonia did in that cranial ventral region. All right, then you may have an embolic pneumonia. This happens a lot, especially in young calves that have liver abscesses. So for instance, if you had an umbilical infection and then that umbilical infection traveled to the liver, then it can enter the bloodstream and it can go into the heart, and then it can be distributed into the lung, where it makes all these random focuses of inflammation. All right. Similarly, you can get granulomatous pneumonia, and usually we see that like with diseases like tuberculosis, when it's kind of a long-term infection where your body is trying to wall it off and preserve it. Although this was made for cattle, we rarely ever see we rarely ever see tumors. They can happen, but they're just less common. And then we have a few other ones like verminous pneumonia. So verminous being like lungworms. So lungworms tend to be in these caudal parts of the lung lobe right here. And the way I identify them as a pathologist, because they're really subtle, you kind of rub your hands over that part of the lung and you might feel a little nodule. You may not see it, but you may be able to be like, oh, that feels a little bit more dense in that region. And so that's the more common place to find lungworm in any species, but particularly ruminants. And then last, we have aspiration pneumonia. That tends to be more up in this cranial, but particularly in the middle lung lobes, just because when you aspirate, you kind of take that deep breath in, or you're choking, and that the feed material tends to not fall out too early, but it makes it down into that middle lung lobe. And the other thing that can look a lot like or be concerning that you may not know whether it's a lesion or not is congestion. So like when the animal is on its side for a really long time, the blood will pool on one side of the lung, and you may not be able to tell what, like if that is something to be concerned about or not concerned about. All right, so what are some of those pictures? All right, so as promised, we have some graphic pictures. So we were showing it to you kind of looking at it from the back, in those last distribution pictures, this is looking at it from the side, which is the typical way to open up an animal, like Gaby showed you in that first kind of video of how to do a necropsy. You can see that this part of the lung that's really dark red, and this is the affected part of the lung, versus this kind of pink lung that's kind of standing up a little bit more is the normal lung. You can see that it's at the front part. So the head is over here, and then you're coming into that top part of the lung. So it's in the front and bottom part of the lung. So remember, those bacteria come in and then they fall out of the airstream into these lungs. So this is like a bacterial bronchopneumonia. This is what it might look like on cut section. Here's the normal part of the lung. And then this part down here is really firm. And if you were to just cut off a small section of this lung right here, it would actually sink in formalin because there's no air in there. So if it has this part attached to it, it will actually float. But if you only cut this part off, it'll sink. And you can see right here where this arrow is and where I'm pointing to, this is an airway that has a lot of what we call pus in it, right? So that those are neutrophils, part of our immune system that are trying to fight off the bacteria. To give you an example of that pleuropneumonia or fibrinous bronchopneumonia that happens with Mannheimia. So this is fibrin that's sitting on the surface of the lung. So it's sitting all over the lung right here. And then this is that consolidated portion that's similar to this over here. Just a different look at it. On cut section, you can see these are all the little septae that separate each of the little lobules of the lung. And they have a lot of fibrin and edema separating them. Then we have that interstitial pneumonia that I mentioned. See, the lung really stands up. It doesn't collapse. See how you kind of have more kind of peaks and valleys a little bit here in the normal lung up on top. This one really stands up like styrofoam. And then you can even see where these arrows are pointing to rib impressions because it's so big that it actually pushes up against the ribs. So that would be more of your viral pneumonia. And then you have the embolic pneumonia, like from a hepatic abscess. All right, some other things that you might see when you're opening up the animal, you might see weird sections of lung that look like this. These are actually airways. So remember how we talked about all of these branching airways up here? So these are all those branching airways that are filled with pus, and it's what we call bronchiectasis or bronchiectatic airways. And then you have consolidation of the lung here. So it's good to take a part of this where you have the bronchiectasis, but it's also really important, as Gaby mentioned, to take a part of the consolidated tissue up here too. All right, then the other thing you might see, so this is fibrinous rhinitis. So you can see that there's all that fibrin, like we talked about on the surface of the lung. Well, here is some that's kind of, this is a head that has been split down the middle. These are your nasal turbinates. They section out your nose into different sections. And lining the surface of this kind of nasal cavity is fibrin that extends down into the esophageal region right here. So this would lead down to the esophagus and the rumen. That all has fibrin on it. In addition, this is the trachea that has been opened up, and it also has a bunch of fibrin, or what we call a pseudomembrane. It looks like it has something lining the whole surface. So this is what happens there. This is what can happen in diseases like IBR. So that bovine herpes virus 1 can cause bovine rhinotracheitis, right? So rhinotracheitis. All right, some of the other things that you might experience in your herd are what we call necrotic laryngitis. This is really common in calves. So as they get bacterial infections, this is the epiglottis. This separates the esophagus from the airway to kind of prevent us from aspirating any food. At that region, if you get swelling of of the tissues surrounding that, they hit against one another. And when they hit against one another, they abrade the surface. So they slough off some of the surface of the, that's protective, what we call the mucosa. And then that allows for that bacteria, the commensal bacteria, the bacteria that normally live there, to overgrow and cause disease. Then you can imagine if this is a bunch of bacteria and inflammatory cells and fibrin, it's really easy for pieces of this to break off and then go down into the lung, right? So that can also lead to pneumonia, and it can lead to systemic disease that can kill the animal. All right, and then lastly, on our trip down bovine respiratory disease, we have this is kind of one that is a toxicity. So fog fever, you might have heard about it before. This is when you have a cow that is moved from a pasture that's kind of dry grasses onto a really lush pasture. Or what we've seen a lot of this year is that a lot of the grasses, because we had such a bad drought last year, a lot of the grasses were brown. And then with the first big storm, we had that like monsoon storm here in California. With that first storm, all of the grasses came back to life and were really lush and green. And that caused fog fever in a lot of our herds. And what it is the lung looks emphysematous, like it looks like all this bubble wrapping, and it feels that way. If you run your hands over it, it's all of this gas production throughout the tissue because all of the air spaces die because that lush forage, when you get a lot of lush forage into the rumen before the rumen has a chance to acclimate to the new feed source, it produces a toxic compound that targets the lung. So it's one thing that we've been battling with, especially this year.
Dr. Watson?
Yeah.
We did have one question come in about fog fever. Someone was wondering if, aside from lush pasture, could alfalfa do that as well?
So usually the problem that we think about with alfalfa is more of what we call bloat, like so a frothy bloat. Usually it's the actual tryptophan, like within the conversion of the tryptophan within the lush fresh pasture rather than dried alfalfa. Because alfalfa is a little bit more stemmy. So we worry about the protein content in alfalfa causing rumen bloat rather than fog fever. But I'm sure that there could be other reported cases. All right, so the most commonly affected sites in blackleg were all of the above. So here is a schematic that came from a recent review article by one of the California Animal Health and Food Safety pathologists. His name is Francisco Uzal. He's actually the top veterinarian who studies this disease, and he knows the most about it. You can see that it happens in those hind limbs within the diaphragm, the heart, and the tongue. And these are representative examples of what you'll see. So in that hind limb, you'll see that there's an incredible amount of hemorrhage. And if you haven't seen necrosis, and necrosis just means death of tissue, that's what this looks like. So the tissue is dead and it has a lot of blood that's outside of vessels in it. A lot of times it will have, like, if you look over here at picture C, it'll look like, remember that lung looked like, it was what we call emphysematous. It looked like bubble wrap. So the muscle can look like that too, because part of what the bacteria does is produce a lot of gas in this anaerobic or low oxygen environment. The heart will have similar changes. You can see that there's necrosis and hemorrhage within the wall. And then here's a picture of the tongue. So the muscle in the tongue will look similar as well. So all of this is caused, blackleg is caused by a Clostridium species, which is a big fat bacillus, so a big rod-shaped bacteria. And it's the specific one that causes blackleg is Clostridium shoveii. And it really targets muscles, both heart and skeletal muscle, which skeletal muscle makes up the tongue, the diaphragm, and those hind limb quarters. All right, so as I mentioned, Clostridium shoveii is the cause of blackleg, and it can be prevented by vaccination. However, vaccination is not 100% effective. We still see sporadic cases where vaccination failed or outbreaks, whether that's because it's a more virulent bacteria than what they were vaccinated for, if the vaccination failed because maybe it wasn't stored correctly, maybe the time between vaccinations was too long, maybe they missed that series. All of those things can happen in the vaccination, and we still see cases even in a vaccinated herd. And then this is a fetus that was reported. So the dam had blackleg in the hind limb quarters, and then it went systemic and it caused abortion because then the fetus was also affected with it. So these are tissues in the fetus. You can see here are those bacteria stained with a special stain. You can see the rod-shaped bacteria within the heart of the fetus here. So how does blackleg work? So you're probably wondering, like, where do these bacteria come from and how do they cause disease? So bacteriospores are within the soil. So they live for a long time within the soil. And when the ruminants are grazing or foraging, the spores are absorbed into the intestine with the food. Then they go into the bloodstream. And once they're in the bloodstream, they travel throughout the body, but they have a propensity to go dormant within muscle, whether that's cardiac muscle or skeletal muscle. Then when there's a site of injury, usually it's like if you're running them through a chute, or if they get kicked by another animal, or if they run into a fence or a gate, or if the gate closes on them, that injury causes a hematoma, so a bruise. And that causes an anaerobic or a low oxygen environment, but the spores then start to germinate. They start to make more bacteria. They start to propagate. When they propagate, they make a toxin, and that is what causes all of the local damage within the tissue. Then that toxin can go into the bloodstream and cause an enterotoxemia. That just means that you have toxin throughout the bloodstream, and that is what causes death. So even though you have a local area of injury, it's the toxins that are produced by the bacteria that cause death in the animal. All right. Any questions about blackleg? All right, hearing none, I'm going to go ahead and move on. All right, we'll talk a little bit about the heart and some of the diseases.
So there was one question that just came up. What are the lead-up symptoms?
So usually there are no lead-up symptoms. That's what makes it so difficult, is a lot of times these animals will just drop dead. And it's part of it is because cattle are extremely stoic. Because they're a prey species, because they're predators that are always looking for the weakest within the herd, they do a good job of hiding disease until it's too much for them. So oftentimes they will just be found dead. But it can, they can have limping or, but it tends to be that they drop dead before anything else.
And that answered the other question about how fast does it kill?
Gotcha. All right. So some of the diseases that affect the heart. So these are like cross sections through the heart. Sometimes it's hard to picture it, but this is kind of the apex of the heart here. That's just the like rounded part at the bottom, and it's been cut off on both sides to make a big slice apart. And you can see kind of like what we were looking at with the black leg. You can see that this is kind of more normal colored heart. It's kind of my cursor is kind of blending in with the tissue, but you can see it here. And then this is the more abnormal heart. This is like a papillary muscle. That's like a big muscle that helps close the valves of the heart. So you can see that this one is white, more white. This one, you kind of have both areas of hemorrhage, all of this red over here, as well as the necrosis. And then this one is like that pleuropneumonia. This is all fibrin that's on the surface of the heart. So you can think about it as like, if you look at this picture down here, this is fibrin that's totally wrapping around that apex of the heart that you're looking at right there. So this here is white muscle disease. The only way you can tell the difference between this one and this one, which is bacterial myocarditis from either Histophilus somni or that black leg agent, Clostridium shovii, is histologically. So histologically, like when Gaby talked about looking at it under a microscope, the white muscle disease, which is a deficiency in vitamin E and selenium, has a lot of mineralization. So this is a special stain that shows all of the mineralization. So the white in this case correlates to mineralization within the tissues. Versus in here, you have a lot of inflammation. So these are neutrophils. And then within vessels, you have bacteria. So these arrows and the blue in the tissue here are pointing to tons of bacteria that have lodged within those vessels, because Histophilus somni likes to bind to vessel walls and cause what we call hypoxia, where blood can't get through. And so the tissue dies because it's not oxygenated. Versus hardware disease. Hardware disease is caused by the cattle ingesting sharp objects. So while they're out there grazing all of the hopefully not too lush forage, then they may pick up some metal pieces, and then those metal pieces fall out into what's called the reticulum, which is the first part. It's one of the first chambers of the rumen. And then they poke through the rumen, because the rumen is down here. It pokes through up into the pericardial sac and causes bacteria to lodge all around the heart and cause inflammation. It is amazing that the cows survive as long as they do with all of this fibrin around their heart. So that goes again to the fact that they are super stoic. They can have significant disease before they just drop dead. So sometimes it seems like there's an outbreak of hardware disease. And really it may be that these animals had a problem a long time ago, and they've just been holding it off for as long as they can. And then they kind of all drop at similar times. One of the ways that this is prevented now is by feeding magnets to cattle in order to try to get all of those metal pieces oriented kind of in the same direction, in hopes that they won't poke through and cause disease. But we still do see that disease. So because somebody asked about Histophilus somni, I wanted to talk a little bit about histophillosis. That was one of those causes of the heart disease. And you can see one of the well-known things that it causes is what's called thromboembolic meningoencephalitis. That's just a very fancy word for all of these bacterial emboli within vessels throughout the brain. So similar to this picture right here, see all of these bacteria within the vessel. They can also go all throughout the brain, and then the animal can present neurologic, and it causes a lot of other disease because it travels throughout the blood system. So it's a normal commensal in the respiratory and repro tracts. And it's when we get that disequilibrium that we talked about with bovine respiratory diseases, that it can move systemically. So bovine viral diarrhea virus, it's caused, like the name implies, by a virus. It's a little redundant. So a cow that has never had a BVD infection before is considered naive. When a naive cow is infected with the virus, they get infected for a few weeks while their immune system learns how to fight it off. These animals are called transiently infected cattle. And a transiently infected cattle means that the infection won't stick around. So if you test the blood of animals that have recovered from being transiently infected from BVD virus, then they will have antibodies showing immunity to the virus. All right. And then eventually they usually recover. So typically a transiently infected cow doesn't stick around, and then they become an immune cow, and they may have BVD antibodies in their milk and their blood. All right. So where do transient infections come from? BVD doesn't spread efficiently from a transiently infected cattle to a naive cattle. So because the transiently infected cattle, they don't excrete enough of the virus. So you can kind of think about it in today's terms, like with coronavirus. The reason why they wanted people to get vaccinated is because even if a vaccinated person comes into contact with somebody who's secreting coronavirus, then they are much less likely to shed a lot of virus to infect other people. That being said, my kids are vaccinated, and right now we're quarantining because they both came down with COVID during spring break. It is what it is. But that's the kind of thought process behind it, okay? And so how do we get a transient? How do we actually get something that makes a transiently infected cattle? It's when a naive cattle comes in contact with a PI cattle. So a PI cattle is a persistently infected cow. This is a cow that sheds a lot of virus all the time. So how did we get a persistently infected cow? That was when mama cow was pregnant and came in contact with a persistently infected cow, and that created this persistently infected cow that will be birthed and continue to spread the disease in the herd. So these persistently infected cows, unlike the transiently infected cows, shed a lot of virus, and they shed a lot of virus all the time. And that's what causes disease. So usually it happens within a certain time during pregnancy. That's like within the 40 to 125 days, approximately. The numbers range depending on the resource. Before that, you'll probably have embryonic deaths. And after that, you may have weak or stunted calves, birth defects, or stillbirths. But once you have a birth of a persistently infected animal, then that animal will shed BVD for life. All right, so there's two different types of disease that happen. The disease that you might see on your ranch that where the animal dies from the disease is from mucosal disease, and that is from when this persistently infected cow gets exposed to another BVD strain that it isn't shedding. It can cause severe fatal disease where you get ulcers in a lot of the tissues, or severe acute BVD. And that's what we think about with the bovine viral diarrhea virus. Like that's where the diarrhea comes from, because they get severe diarrhea and then they can get immune suppression. So then their immune system doesn't work very well and they die of other diseases. So this is what that mucosal disease looks like grossly. You can get ulcers at the coronary band down here, as well as along the digits. You get ulcers and erosions in the mouth. You can get respiratory disease. So this is all of that nasal discharge. Here, this is nice because you can see that this is the roof of the mouth, and you can see these white spots are all these ulcers that are all over the roof of the mouth. This is all like fibrin and ulcers along the esophagus. And then you have ulcers here within the abomasum, so within the stomach. And then that's similar to the clinical findings in acute BVD. So it likes to target the lymphoid tissue. So in the intestines, you might see these ulcers, and this is usually at what we call a Peyer's patch. You can kind of see it on the outside of the intestines right here. And this is lymphoid tissue. These are lymphocytes. That's part of your immune system. So we talked about neutrophils earlier. Those are our early immune system. And then lymphocytes are our later immune system. They're part of our white blood cells, and it causes necrosis of those tissues within the intestine. So those are some of the things that you might see with BVD.
Dr. Watson, we do have a question.
Yeah.
What is the percentage of cattle that are considered to be PI positive?
So in all of California, so it's hard, right? Because we're like, it probably varies a lot throughout the state, just depending on the different management techniques that are happening on the farm. And I don't know, Gaby, if you have a better feel for what those numbers are.
No, I mean, I don't have a specific number either. I was going to go in the, the same direction. It just depends. I mean, if you have a closed herd and your cattle are vaccinated, you probably have zero. I mean, hopefully you have zero. But it, the percentage of cattle that, that have just gone through, have just been trucked from Idaho to California and are, spending time in several livestock markets during that journey is probably a little bit higher because they're stressed. They've been exposed to a number of different herds. So it really depends on those risk factors. Have they been commingled, or how stressed are they? That kind of, depends on whether they get infected, with how likely they are to be infected. They exist. I can't give you an exact number, but that's why it is extremely important to make sure that you do not introduce one of those into your herd. I mean, it's a small percentage. I'm sure it's not like 10%. I don't know. I mean, it's, I don't want to give a number because I really don't know it, but I think it's a small number. But again, a single one can really do a lot of damage.
Yeah, and I think that the other really hard thing about BVD is that there's still so much unknown about the actual transmission, in the sense that there can be a PI animal in the herd, and you would think that based on how much it's shedding, that everyone would be immune after a certain amount of time, but for some reason there are always still naive animals that haven't been exposed and come up, and the PI animal can live for a long time. So they're usually poor doers, and they usually die before their kind of normal natural lifespan, but they can live for a long time and infect a lot of animals before that happens.
Yeah, and the problem with the PI, with the introduction of a PI into a group, is that then you perpetuate it. That pregnant animals then have calves that are also PIs, and et cetera, et cetera.
There was a question about testing for it.
Oh yeah, so that's a great question. So at CAHFS, we offer a lot of different tests. Most of what people are interested in are testing for PI animals, and the easiest way to do that. So because a PI animal, when it's in utero, it doesn't have an immune system yet, and that 40 to 120 days is when it's starting to recognize itself, and so it recognizes the virus as itself. So it's in all of the tissues, including the ear. So the easiest thing to do is to take a little ear notch and send it into the lab, and then that can be tested for whether that animal is a PI animal. You can also, so if you draw blood, it also will not have antibodies to BVD, and that is because it never mounted an immune response to BVD because it never recognizes it as a foreign substance. But if you want to confirm that it does have BVD, you do that ear notch and do the testing with that. At Necropsy, we test other tissues. So there are other tissues like the spleen that we can collect at Necropsy to see if it's in there with molecular tests.
Thank you to Dr. Watson and Dr. Maier for sharing with us common cases and resources at the California Animal Health and Food Safety Laboratory. During the video, there were references to other webinars. You can find recordings of those sessions on this YouTube channel. Thank you for watching this webinar recording that was brought to you by the University of California Cooperative Extension and University of California, Davis Veterinary Medicine, and was co-hosted by University of California Cooperative Extension Advisors Tracy Schohr, Grace Woodmansee, Rebecca Ozeran, and specialist Dr. Gaby Maier.